I'm going to go through some mitosis notes with you guys, like what it is, the different phases, and what's happening in all of the phases. So there's three main stages of the cell cycle. We have the very first stage is interphase. This is where the cell grows, it carries out all the functions, makes copies of the DNA to prepare for the next stage. And it's divided into three substages, which we filled out on your chart. So I'll kind of skim through these. Mitosis, it's a stage that has four substages, and we'll go through those in a bit. And near the end of mitosis, cytokinesis happens. And then cytokinesis, that's where the cytoplasm divides and it creates the actual new cell. So most cells go through this whole cell cycle an average of 12 to 24 hours, so pretty quick. And that is continuously happening. So the three substages are G1, S, and G2. So the first one, G1, this is right after a cell divides, so the very beginning. And the job of the cell in this stage is to grow and to carry out the normal functions that we need and to prepare to replicate. And then the nerve and the muscle cells, they exit the cell cycle here and don't divide anymore. So they kind of follow a little bit different cycle there. The next substage of interphase is the S phase. This is where the cell copies its DNA. And then what's called a chromosome, this is where your genetic material is and where it's passed on from one generation of cells to another. And chromatin, another term, is the actual jelly-like DNA that's in the nucleus. So that's called chromatin. And then the last substage of interphase is G2. This is where the cell is preparing for division, and the proteins that make the different microtubules are made. And the cell starts getting ready for mitosis. So within mitosis, the cell's two copies of DNA separate, and the cell prepares to split into two cells. And the resulting two cells are genetically identical, so they're the exact same. And we'll get into like meiosis next chapter where that's not the case, but for mitosis it is. So in multicellular organisms, mitosis increases the number of cells as a young organism, and then it grows into its adult size. So mitosis is also used to replace damaged cells. So say you get like a cut or a scrape, mitosis works to help heal that with new cells. So there's four main stages of mitosis. You have prophase, which is first, metaphase, anaphase, and telophase. And again, I always tell students, kind of remember just like PMAT, it just takes the first letter of each phase and puts them in order, and it might help you remember the order. If you have another way, that's great. So just breaking these down a little bit, prophase, the very beginning, it's the longest stage of mitosis. Something called chromatin, it condenses into chromosomes. The chromosomes, they look like an X. So these right here in this example would be your chromosomes, where like your genetic material is on. And if we took like a half of a chromosome, so each half of the X is called a sister chromatid. Sister chromatids contain identical copies of DNA. And then that structure in the very center of the chromosome, or very center of the X, where the two sister chromatids are attached, is called the centromere. It's important to make sure that each copy of DNA goes into its own new cell. And then as prophase continues, the nucleolus disappears. So a lot's happening in prophase. So you also have microtubules, and they're like these little spindle fibers everywhere. They form in the cytoplasm. And then the centrioles move to different ends of the cell. This is not happening in plant cells, though. And then something called aster fibers come out of those centrioles. And you'll need to know spindle fibers, centrioles, and those aster fibers, they make up what's called the spindle apparatus. So a lot of terms here to know. What that is, it is important to help pull the chromosomes before cell division, and that envelope disappears and they attach to the sister chromatids of each chromosome to opposite ends of the cell. And here's what prophase looks like. The spindle is forming here. You have centrioles here. Whoops, did not mean to do that. And then the centromere would be the center of the X, and then the X's are obviously the chromosomes. And then the nuclear envelope that surrounds them is shown with that dotted line. And if I do have you do the bag thing to try to create your own, we'll check it with this slide, and I'll just pause it. Just another image there, microscopic image. And then the next phase is metaphase. Sister chromatids are pulled by proteins to the center of the cell. So the chromosomes, the X's are lined up in the center, or I always tell students, like, remember the chromosomes are lined up in the middle, like middle metaphase. They both start with M. And it's one of the shortest stages of all of mitosis. And here's what it looks like. These are, again, the spindles and the centromeres, the chromosomes, all of that. The nuclear envelope disappears. Now, I may have you do this with the bag, possibly, so I'll pause it if we do that. And that's what yours would look like with my materials. Okay, the next phase is anaphase. So the spindle apparatus is starting to shorten here, pulling on the centromere. They are pulled apart. And I always tell students, remember anaphase and apart both start with A. Might help you remember this phase. And here's what it looks like. You can see the chromosomes are being pulled apart, starting to create that new or two new cells here. And that's what yours should kind of look like if you use the materials. And then the last phase of mitosis is telophase. So this is the last stage. The sister chromatids, they arrive at the ends of the cell and they decondense. And then in both new cells, the nucleolus and nuclear envelope reappear. And then the spindle apparatus breaks apart. And that's what it's starting to look like. You got your nuclear envelopes reforming in both cells. And that's what your bag material should look like. And then the last part is called cytoplasm. It divides, resulting in two identical cells. So this is the actual division part. In animal cells, the cytoplasm is pinched, forming two cells. In plants, the cell plate forms between the two nuclei, and then a cell wall forms on both sides of the cell plate, still making two identical cells. And that's just showing a picture of it. And this is plant. You can see the cell wall there. And that should be where we complete this